In this video, we're going to look at the separation technique of chromatography. This comes in C1. In C1, I am going to cover the basics of chromatography and the way in which it is used for a separation technique. You will also meet chromatography in C8. In C8, it is a required practical, and you will need to watch the linked video in C8 on the YouTube channel, which has the further details. So in this case, we're looking at chromatography as a method of separating inks or dyes. Now this can be inks from pens or food dyes. Usually what we do is we take a piece of chromatography paper, like so, and we draw a line along the bottom. This should be drawn in pencil to prevent it from running when you do the experiment. This line is called the baseline, and it is there for us to place our dots on so that we can then compare the different colours. You place your dots on the line and then place them into water. Now the key thing here is that the pen should not go into the water. In order for it to run, the water should be below the line. As the water is absorbed by the paper, then the pens run. Now they do this because they are water soluble. If I completed this practical using a biro, which is not water soluble, then the ink wouldn't run and you would just be left with a dot. In industry, they use this to compare different dyes. So usually they will have 2 or 3 different dots and they will compare the dyes to see what they are made of. As it runs, you can see that it shows that the black pen is actually made up of blue ink and some different shades of blue in order to make the black. As I said, this will only work with substances that are soluble in water. If you wish to do it with a biro, then you would need to change your substance here for ethanol, which would then allow that ethanol to run the biro, which is insoluble in water. Key errors with this practical tend to be to do with the drawing of the line and also with the insolubility of the substance that you're trying to run.